Hello, everyone, and welcome to another episode of Not So Serious Science, where we explain science in a way that even your neighbors will understand. I'm your host, Emmanuel, and with me is my co-host, Pat. Today is my extreme pleasure to introduce a very good friend of mine, as well as an amazing scientist. His name is Dr. Ted Lakowski. He is currently a professor at the University of Manitoba College of Pharmacy. He will be explaining to us some of his research interests, which includes the influence of disease states and drugs on epigenetic modifications. Welcome, Ted. Can you kind of briefly describe what your research interests are right now?
Okay, sure. Probably the best thing for me to do is to explain what epigenetics is, because that's the general area that I work in. And so most people are aware that you have DNA in your cells. And the DNA is like a blueprint, how to make more cells, more of you, that kind of thing. It's a genetic blueprint. And we've known about this blueprint for quite some time and what the sequence is. Scientists would refer to this as the human genome. And originally, when they figured out what these blueprints were, which are a series of 20,000 what we call genes, I think a lot of people thought that suddenly this was going to translate into solving a lot of diseases, generating a lot of drugs, and it hasn't really turned out to be the way that they initially thought it was. What the blueprints or the genes don't tell you is how much of that blueprint or gene is made. And scientists call this gene expression. So you have the instructions to make the gene, which are the blueprint, but then you have the question of how much of that gene is made. And you probably have heard something crazy like we share in terms of our genetics 97% with chimpanzees.
Right.
And so a lot of people thought like, oh, this is great. We're just like the animals. We're so fantastic. What you're not seeing in that is that, yes, we share 97% of our genetic material with chimpanzees. But what you're not seeing is that the amount of each one of those genes that's produced is different. And that's probably actually what accounts for most of the difference between humans and chimpanzees. So if you were to just look at the blueprints, if you were just to look at the genes, you would get the false impression that we were 3, I can't remember, it always changes, percent similar to chimpanzees, but that's telling almost half of the story actually.
Let's say like from that 97% of genes, is it the fact that they're being produced at each gene of that 90%, 7% is produced at different variety of amounts, or is it because some of them actually are turned off and they don't even produce anything?
It's both. Okay. Yeah. And actually you can go and look at those genes, many of the genes that we have that are similar between chimpanzees and humans. And you can see some of them are completely turned off in chimpanzees and they're turned on in humans and vice versa, right? So what's really cool, if you think humans and chimps are cool, what's really cool is the similarity between birds and dinosaurs. This will blow your mind because birds have many genes that we suspect are actually they inherited from dinosaurs. And now there are people in labs who make Franken-birds, basically. That's the best way we can say. They turn on dinosaur genes in birds. And in some cases, they've grown teeth in birds. With birds, of course, don't have teeth. They have beaks and stuff like that. This is usually in embryonic stages, right? That you can do this. They don't survive into adulthood.
That's crazy you mentioned that because Emmanuel and I just learned that not too long ago. We interviewed Dr. Corin Sullivan from University of Alberta, who's a paleontologist, and he was telling us that birds are actually dinosaurs. So we have living dinosaurs with us today. I mean, you can trace it back. Yeah. So yeah, one thing I didn't know is that they're actually creating Franken-birds. So that's kind of scary.
Yeah. So I mean, it's funny. I told my students that if you wrote Jurassic Park now, you would say that you were turning on. You would say that we would turn on dinosaur genes in birds and produce something that would be not a dinosaur, but would be something like a bird dinosaur hybrid. But I think that's just an example of what's going on with epigenetics. So epigenetics basically is the study of inherited changes or inherited gene expression levels. So we inherit from our parents the fact that some of these genes are turned off and turned on. And this, in combination with our genetic code, is what makes us what we are. And I don't know, probably other people have come up with this, but when I talk casually with people, I call this the second dimension of your genes. The first dimension is the actual DNA sequence, which gives the codes, the instructions for making those molecules, the proteins and whatever. The second dimension is how much is being produced. And actually, sometimes I'll say there's even a third dimension, which is when you make those proteins or those molecules, they have a certain amount of activity level associated with them. And that's the other thing that's what I would call the third dimension of the human genome. And so what my lab is doing is looking at medications and producing medications that intervene in this whole complex. And of course, the simplest and most obvious disease target would be cancer. But it turns out that epigenetic causes are being found in a number of different diseases, and some of them, including possibly multiple sclerosis and a bunch of other diseases. There are some people who think now that the long-term consequences, the hardening of the arteries that you get from coronary artery disease, may actually have an epigenetic cause. The number of diseases that we are now beginning to think possibly have an epigenetic origin is expanding.
What about in terms of, like, for example, infectious diseases like COVID then? Does this play a role in susceptibility?
Certainly it's possible. I can say from my research into this and the way that people respond to drugs, then that certainly has a potential epigenetic component. Does that mean then that the people who are susceptible to dying from COVID, do they have some kind of epigenetic component? I can't tell you that, obviously, because we just don't know. But certainly we know that the way that people respond to medications, there is an epigenetic component to it. And we know that some of the drugs, for example, I think that we're fairly certain now, in the right context, if you're being ventilated, that dexamethasone will reduce your chances of dying from COVID by, I think it's about 25 to 30%. And we know for a fact that what dexamethasone does is it changes gene expression. That's the way that it works. So it changes the amounts of these proteins and things that are, and we've known that for a very long time. So certainly there's a possible epigenetic component to the way that some of these drugs work.
So Ted, can you confirm something for me? So each gene produces a bunch of proteins, right? Not just one protein.
Potentially, yeah. Something called gene splicing. Yeah, which means that basically you're taking segments and you're putting them together to make different versions of the same gene sometimes.
Okay. And so is the concept about disease where, you know, what you're looking at is some genes may be turned on or turned off, or some genes, the proteins they produce may not be, the norm as like a regular person, or there might be a modification of the protein that gene produces, like all those come into play?
Yeah, certainly. You get different variants of each gene that you can produce, and you find that some of them, it could be something as simple that it's targeted to a different part of the cell. Whereas the regular gene just goes anywhere in the cell, and then the variant version only goes into a certain compartment inside of the cell. But it can get much more complicated than that. And I'm just giving us a very facile example.
In your lab, for example, how do you develop a therapy or therapeutics against cancer in terms of epigenetics? Because, I mean, cancer cells are basically mutated or different forms of cells already in our body. So how do you utilize that to hopefully develop a therapy?
We know now that there are some cancers that are caused by aberrant epigenetic mechanisms. We in particular look at one type of leukemia called mixed lineage leukemia, although we're looking at gastric cancer and other diseases. If you just look at something like gastric cancer, we know that there's a subset of gastric cancer, so stomach cancer, that's caused by low amounts of a subset of genes that are called tumor suppressors. And tumor suppressors, should be obvious what they do. They suppress the formation of tumors. So you can imagine that if you don't produce enough of something that suppresses a tumor, then you're more likely to develop a tumor. What my lab is trying to do is be really specific about it. So if the disease is caused by low expression of a tumor suppressor, a particular tumor suppressor, I'm thinking of one called P16, then how do you increase the expression of that tumor suppressor in order to fight the disease? We're trying to be specific on the gene level. So can we turn on that gene? There are a lot of processes to shut down expression, and there are many different methods to do it, but there aren't a lot of very good methods to turn expression back on. And sometimes that's something that you need to do when you're trying to treat a particular type of cancer. So gastric cancer is one example that we look at. We look at another example of a leukemia, like I said, called mixed lineage leukemia, that the cause of that is from chemical modifications to proteins called histones. And histones are important for packaging DNA. So you can imagine we have a lot of DNA in our cells, and there has to be a way of storing it and organizing it inside of the cell. The interesting thing about the way that the DNA is organized and stored inside of the cell is it's not just to package the DNA neatly inside of the cell. It also at the same time is involved in controlling how much of those genes are expressed at the same time. So by controlling the packaging, you also control the expression levels as well. We're trying to be gene specific and regulate that packaging so that we can turn on or turn off the expression of a gene accordingly, depending on if it causes cancer or not.
So are you saying like leukemia and gastric cancers, is it kind of like breast cancer where it could potentially be passed on? It's like hereditary?
For sure.
Are these
absolutely
type of diseases, that you're...
Certainly possible, yeah. Certainly possible.
Okay.
You got to remember, and if I'm just thinking about the mixed lineage leukemia, there are two kinds. There's the kind that you acquire as an adult, and there's the kind that's acquired in childhood. And I think the childhood one would be inherited. The one that you get as an adult is not.
So what makes a disease more hereditary than others? Like what makes that gene stick and others, don't?
There isn't really a way for me to answer that with a single answer. In general, if you're talking about what can be passed on, it can't be very detrimental up until you're past or in the middle of your reproductive years. This is why diabetes, which is, very detrimental to your health, exists in the population. We've known about it since Egyptian times. Egyptians were writing about diabetes in the, thousands of years ago.
Oh, really?
It exists because you can live to 13, 14 years old with diabetes, sometimes even older, without any kind of treatment. So you can reproduce and then it gets passed on. I mean, it's basically exactly what you would think with evolution. It has to be something that's not so detrimental to the organism that it isn't capable of reproducing. If it's an inherited modification that changes the expression of a gene that causes you to die in utero, then you don't get to pass that on, right? There are probably an uncountable number of diseases that we don't really know a lot about because, you get a miscarriage in the first trimester or something like that, instead of, giving birth to a child who then develops a disease.
Yeah, so it seems like a lot of these epigenetic changes, it's basically a form of evolution in that sense, eh? It's kind of either, I guess, finding ways to adapt to pass that gene on, either for good reason or even a bad reason. Like you said, potentially having the miscarriage in this sense may be a good thing to stop it from being carried on further
to the next generation
and so forth. What are your thoughts then with cancer then? I mean, cancer is getting passed on, generation to generation. Is it just a way of evolution where eventually one day you're just hoping that that gene will just kind of be out of the gene pool? Or
the genes that are mutated that cause cancer, we want those out of the gene pool. And I should state here that a lot of the time, what we're talking about is a disease that is caused in adulthood. So that still is technically an epigenetic origin because the cells are passing on that inherited gene expression to the cancer cells are passing it on. So we can't think only of sexual reproduction. We have to think about the replication of the cells. That's still an epigenetic mechanism. If the cancer cells are passing along that mutation or that change in expression to other cells, then it is still an epigenetic etiology. It's just not in the sense of like what we're thinking right now in terms of generational reproduction. We're talking about replication of the cells. And of course, the most obvious and dangerous thing that you can think of is a tumor that keeps growing. or cancerous cells that are in your blood that keep growing, and they carry on this epigenetic modification from generation to generation of the cells in the body.
So mapping out the human genome was a huge leap in your field.
Yep, absolutely.
In your opinion, what is the next big thing that needs to happen to really advance?
I think when the human genome was first mapped, a lot of people thought, well, this is the template, right? These are the instructions. I mean, what we found since is that they're tiny modifications that you get in 1% or greater of the population. There is this template sequence, but there's a lot of variation in it in the human genome. And those certainly can be a result of disease. Also an issue with the way that people handle medications as well. With the human genome, we could sequence it. And then we had this thing called the genetic code, which, undergrad students, sometimes even high school students, will get a genetic code, and they'll look at a piece of DNA, and then you'll look at your genetic code, and you can tell what is the sequence of the protein that this gene is supposed to encode. And it's very straightforward. Anyone with two pieces of paper and a little bit of knowledge can actually take a gene and tell you what is the protein sequence. And in fact, there's a lot of programs now where you'll just feed in that sequence and it will tell you what the protein is. And in some cases, we know what the protein does. With epigenetics,
I'm sure there's an app for that somewhere.
Oh, there's definitely an app for it. The problem with epigenetics is that there are so many competing mechanisms going on, and it's way more complicated than we thought it was. Initially, when people started looking at this, they thought that there would be a similar sort of thing to the genetic code, where there would be a little piece of paper where you could say definitely what was going on. It is way more complicated than the genetic code. The epigenetics, in terms of the way that it works and controls expression, is hugely more complicated. And right now, there doesn't seem to be a very easy way for us to say that we're getting a certain amount of expression of the gene, or the gene is turned off, or the gene isn't turned off, or something like that. Whereas the genetic code was very simple and elegant almost, epigenetics is way more complicated. The thing is that, just like I said that we're different from chimpanzees, remember, you have different cells in your body. Almost every one of those cells has, within limits, has the same genetic code inside of them. So why do you get all these different cells? Well, it's because you have different expression levels that are being produced, different amounts of these genes that are being produced. So each cell is doing that. And then also you have the additional complication that throughout the lifetime of the cell and throughout the cycle of the cell, because the cells will go through cycles as they reproduce, they will vary the amounts of the genes that are being produced. Because sometimes you need genes because if you're a cell that's going to divide, sometimes you need to turn genes on because you're dividing. And then sometimes you need to turn them off because you're not dividing anymore. It's much more complicated than the genetic code. And I think that's part of the problem, is that it is so much vastly more complex. And all we've been doing so far is been finding out, like, wow, it's more complex than we thought. And then every year it's like, wow, it's even more complex than we thought like a year ago. It just keeps getting more and more complex. And that's part of the problem. So it doesn't look like we're going to have a single sort of mechanism where we can kind of look at a couple of different markers and then go, the cell's producing, this level of expression of all these genes. It doesn't look like that is going to happen. It looks like it's going to be very complex. And if there's somebody who can simplify all this for everyone, then I would also like to hear about it, because that would be fantastic.
What's the direction the entire field is heading right now? More towards drug discovery, trying to develop drugs that can, you take a pill and hopefully it'll automatically edit genes in your body?
Well, right now, the idea of taking pills is just to look at those genes that are, by their levels of expression, the amounts that are being produced, are causing diseases. It's kind of an old sort of way of looking at things. We'll try to identify a single target that's this cause, and then develop a small molecule drug to target that single cause. The problem with that is that the enzymes or the proteins that are controlling these epigenetic mechanisms, they work at a number of different places throughout the genome. They control the expression of many, many, many genes. But if maybe a handful of genes or even one gene is involved in causing the disease, then when you try to target it by targeting those mechanisms, you're targeting all the same mechanism where it's doing what it's normally should be doing in the cell. And this is part of the problem that my lab kind of discovered, that when we looked at these small molecules that were being developed by companies, in many cases to treat cancer, what we found was that, yes, it would fight the mechanism that was causing the cancer. But when you looked at other genes in the body, you'd see many other genes changing in expression. And what we found was that it was really very complicated from the point of view that sometimes the amounts of genes that were being changed, those genes were also involved in epigenetic mechanisms. So they were also involved in controlling the expression of other genes. So you get this cascading effect with respect to time, the cell type, and the dose of the drug that you're using. So with one particular medication that we were looking at, we found that the effect that the drug would cause on expression was different in 3 days than what it caused at 6 days, and so on as you go further on. So this makes it really difficult. If you have a drug that's causing, that has different effects depending on how long you've been taking it, that's a little bit of a problem in terms of trying to get some kind of consistent effect out of the drug. There's been a lot of difficulty taking what we know about epigenetics and translating it into a therapeutic so far.
So that sounds very interesting, actually. How duration of drug usage can change over a period of time. So it sounds like the body is always trying to find a way to adapt, really, right? So it's kind of like saying, it recognizes that this drug is causing a change, but the body wants to counteract that change by doing something else. So is there that fear that changing something within our body will potentially lead to another, potentially even more serious change that we're not even aware of?
Right. This is a huge problem. And this is one of the reasons why my lab is focused on trying to confine the effects of these drugs only to particular genes or a gene or genes, because you don't know what happens. If it's working everywhere throughout the genome, you don't know what it's going to potentially do. And we already have experience of something like this. It's not exactly the same. But you are already probably aware that many of the drugs that we use to treat cancer, many of the drugs that are called chemotherapeutics, can also precipitate you having cancer later in life. And we talked about the example of topoisomerase inhibitors potentially later in life causing mixed lineage leukemia in adults. But there are other examples of chemotherapeutics that are used to treat cancer that can precipitate other cancers later. And this is something that you have to be worried about with medication. Is there some kind of downstream effect that you don't know about that may be being caused by the drug that you're treating? Now, unfortunately, when you're talking about that person who's right in front of you at that moment and is dying of cancer, and you're potentially can save their life by giving them the chemotherapeutic, then you have to go with I need to give them the chemotherapeutic. But there's also this other thing that you have in terms of potentially causing some other disease or cancer down the line. We have the same problem with drugs that target epigenetic processes. And it's because their effects are not confined to a single gene or a group of genes, but that would otherwise be known to be causing the disease, but they happen at all of the different genes that these processes work at. So, in many cases, also people get quite sick when they take medications like this, and that could be part of the problem. We don't know, for certain, because it's still kind of in its infancy. But yeah, for sure, the problem of unintended consequences is definitely an issue with drugs that would target epigenetic processes. Certainly, also with cancer in general. Every drug is basically a situation where you're trying to game the system. Yeah. Basically, you're trying to game the system, and you're trying to game a system now that we're dealing with a very much a tip of the iceberg kind of thing. So we don't know what it's going to happen. When we first started looking at these drugs that were being experimentally used to treat mixed lineage leukemia, somehow that drug was working on a totally normal mechanism, which probably shouldn't have been altered. But it was. And that was maybe part of the reason why some of these drugs just haven't been able to to make it to the market. Now that I should say that, there are a bunch of them that are on the market that are being used. In particular, there's a type of classification of drugs called HDAC inhibitors, and those are being used all over the place. But even those cause problems with changing expression of genes that you don't anticipate or even want to have changed. And that's part of the problem. And that's why, again, we're trying to be more specific in terms of how we target the way that those drugs work.
Yeah, I find that a lot of these drugs are intended for treating cancer is because really, cancer, it's so difficult to cure that, using these experimental drugs or these potential drugs as a last resort, right? You may think, it's like, what, we got nothing else that we can give to this patient. Let's try this drug. There's a potential this and this may happen, but this is a risk we're willing to take just because there's nothing else we can try, right? So that's the kind of situation where you would use it. You wouldn't use it again, like to treat myopia, because you're like, well, if it doesn't work, blindness might be worse than, nearsightedness.
Yeah, definitely. Regulatory agencies have different levels of, stringency. And this is true in Canada as it is in the United States. When the disease is more serious, when the consequences are that you're going to die, there are avenues to fast track drugs. There are avenues to fast track drugs that we know even recently for COVID. There are avenues to fast track drugs for HIV, certainly, and there are avenues to fast track drugs for certain very severe bacterial infections. So yeah, you're right in the way that it's easier to try and get your drug approved if you're using it in that context. I completely understand that. I think that's a totally acceptable situation. If you don't really have a lot of options, you kind of have to go with maybe something that's a little bit more dangerous or experimental, potentially. But we're hoping that we're going to be a lot less toxic and a lot more effective doing things the way that we're trying to do it, which is being more specific in terms of targeting the genes that you want to target in particular.
You're doing some really, really cool stuff. Are you looking for new students right now or any new trainees to hire?
You're always looking for talent. You're always looking for talented people, of course, but also driven people. Research is a weird kind of field. I'm always fond of telling people that the only thing worse than doing scientific research is not doing scientific research. It's almost like an addiction almost. In many ways, it consumes all your time and it almost destroys your life in many ways, but you are still compelled to do it. I love discovering something. Yeah. I love being in the lab or being right up against a piece of machinery and having the results sort of come out and thinking to myself, wow, that's not only is that not something I anticipated, but this is not something anyone anticipated. I may be the first person in the world to be observing this kind of result. That is intoxicating. And there's nothing that beats that feeling, that discovering something that's totally new and that nobody knows about, and especially that is going to maybe flip things on its head from a point of view of understanding.
For sure. I think the world needs more people like you that's super enthusiastic, passionate about science, right? So if there's any potential trainees listening in, that are basically sharing the same passion as you do, I would recommend that they reach out to you. They can find you on the College of Pharmacy, University of Manitoba website. Yeah. Right. And then just shoot you an email and then see if there's any potential opportunities. Your lab is constantly expanding. You're funded by, various granting agencies from the Canadian government. So you're doing an amazing job. I mean, kudos to you. And
thank you.
We're definitely looking forward to potentially one day a new drug product from your lab.
I hope so. I hope so. But for right now, we'd be happy to just make baby steps at this point.
Yeah, I think that's the way it is, right? You can't rush it, right? You can't rush through this. No, thank you so much, Ted. I appreciate it.
All right, thank you very much.
Thank you very much, Ted. Take care.
Take care, guys.
Bye-bye.